Okay, we are moving now into section 3, which is on basically RNA and protein synthesis, or the beginning of how proteins are created. And so with that, we first have to cover the structure of RNA because it's a little bit different than DNA. The most obvious difference between DNA and RNA is the sugar involved. With DNA, the name deoxyribonucleic acid comes from the sugar deoxyribose. In RNA, ribonucleic acid, it comes from the sugar ribose instead of the deoxyribose in the DNA. The next thing is that DNA being double-stranded, RNA is just a single-stranded copy of that. It basically is half of the information. If you look over my shoulder, right over here, half of that, just showing the sugar phosphate backbone for one side and one of the nitrogen bases, that would be what a messenger RNA, or what an RNA strand looks like. And the last of which, thymine is not able to bond to the sugar ribose, so it ends up getting replaced by a new nitrogen base called uracil. So it serves the same function, just with RNA, we just have to realize that adenine is going to bond with a uracil to create that complementary strand. So the main reason that we use RNA is because DNA is our important document inside your cells. So that's what allows for your cells to stay alive, creating all of the components for the processes going on inside your cells. So we need to keep the DNA protected, which is why it does not leave the nucleus. And so we still need that information from the DNA, but without the DNA being able to leave the nucleus, we have to make a copy of it. And so it's the same idea with my master copy of worksheets that I give you. So I have a master copy. I go make copies of that to give to you guys for a usable version, so you guys write your answers on it. And then my master copy does not leave my desk. I keep it in the desk so it's protected and not going to get worn down or damaged if you guys would have been using it. So the RNA is kind of the same idea, where it's just a usable copy of whatever information we are using. And so the RNA just uses, or is just used to get that message from the DNA outside the cell to the ribosome, which will make the protein that we need. So there are three, it's types of RNA. Biggest thing is that its function is to create proteins. And so the entire process to create a protein from a DNA strand is called the central dogma. And so with that central dogma, what it includes is DNA getting turned into RNA, getting turned into proteins. And there are two main processes that allow this to happen. The first of which, being right here, is transcription. DNA to RNA is the process of transcription. The second one is translation. RNA creating proteins will be translation. We're going to talk about both of those later on. Transcription in today's notes, and then translation in tomorrow's notes. Just realize there are two processes that allow each of these to happen. So each of those arrows represent one of those processes. In order to get to that point, we need to first figure out what RNA is and what types there are. So there's going to be three different types. The first of which is probably the most common or the most familiar, which is messenger RNA. This would be like the worksheets that I give you. This is the copy of DNA that is the template for how to make proteins. It's kind of the directions of how to make stuff. And so that's going to bring that message to the ribosomes so that the protein can get made. The next of which is called ribosomal RNA, and this is the RNA that is created in the nucleus, just like the other types of RNA, but this is made specifically in the nucleolus. And you may remember from back in chapter 7 that inside the nucleolus is where ribosomes are made. And ribosomes are what will eventually construct the proteins, and they are composed by a large degree of ribosomal RNA. So it's similar to the idea of messenger RNAs as far as how it's created, but the use of it is a little bit different. And the last type is transfer RNA, or tRNA, and these are helping the messenger RNA create that protein. So basically the messenger RNA is the directions or how to make that protein. The transfer RNA is bringing in the raw components or the raw materials to construct that protein strand. Usually they're bringing in amino acids to make that protein strand. So here's just a quick view of each of those types. So all the way on the left here, we have a messenger RNA. Remember, it's single-stranded. So we have a sugar phosphate backbone. Sugar is now ribose instead of deoxyribose. And we have our nitrogen bases, adenine, guanine, uracil. Remember, is replacing thiamine because thiamine cannot attach to this particular sugar. And then cytosine. So everything else remains the same except for our sugar that's involved, and then uracil replaces thiamine. Number 2 is the ribosomal RNA. So this is just an example of a ribosome. So it's made up of several components, all of which being created in the nucleolus. And the third type, the transfer RNA, it's a sequence of genetic information, just like creating that single-strand messenger RNA, except we now have an amino acid that gets attached to the end of this transfer RNA strand. So this amino acid is responsible for helping to make that protein, or the raw materials to create that protein. So 2 processes involved, as we kind of mentioned before, the first of which is going to be transcription. And over here on the right, we have the process of transcription. We have our 2 strands of DNA. They split the same way as DNA replication. Helicase is going to break those hydrogen bonds, open that double helix model up, and a new enzyme called RNA polymerase is going to attach on here and create our messenger RNA strand. So you can see just a single-stranded molecule being created. Still uses complementary base pairing, just like we've seen before. And the key thing here, remember, uracil is going to be in that messenger RNA instead of thiamine, as it was in the DNA strand. So that's the major difference that's going on here. So it creates that single-stranded structure. Obviously, this process is occurring in the nucleus because the DNA is not going to be leaving the nucleus at any point. And so instead of making 2 new strands like DNA replication, we're just going to make that single strand of messenger RNA, which is the usable copy of that DNA. So with that transcription, okay, again, very similar. However, a couple of enzymes are a little bit different. So instead of DNA polymerase, which makes a copy of DNA into another copy of DNA, we use RNA polymerase, which is going to take that DNA strand and convert it into a RNA strand. So the RNA polymerase makes RNA, the DNA polymerase makes DNA. The overall step-by-step is very similar. We're going to have helicase breaking those bonds, opening up the strand of DNA, and the RNA polymerase will attach on, and using complementary base pairing, we can see here the RNA polymerase attaches on, complementary base pairing, adenine with uracil now because thiamine cannot bond to the sugar ribose. Thiamine creates adenine, cytosine, guanine, cytosine, etc. to create that messenger RNA strand. So very similar process to DNA replication. We're only making half of the information as we would have during DNA replication. One thing that is also a little bit different, when you make a copy of DNA during DNA replication, it's just going to make the entire DNA strand so you have full sets of information for the new cells. With RNA, it only needs specific pieces of information in order to create the proteins. So one of those things that's important for this is that there are promoter regions, and the promoter region is basically the RNA polymerase will attach on, it will read those nitrogen bases, and eventually it'll reach a promoter region which tells it to start making the messenger RNA strand. So you can kind of think of that as a green light. So it's traveling along that strand, sees that green light, and now it starts making the copy of messenger RNA. And so the same idea when it finishes, when it gets to the end of that sequence that it needs, there's a stop region, and so it reads the piece of information that tells it to stop. It will release the messenger RNA, and it will continue and look for possibly a new protein or a new messenger RNA to create, or it may release itself from the DNA strand and then be used later on. So kind of a green light, red light, telling when to start and stop to create that strand of messenger RNA. So a couple of vocabulary terms that are going to be important for these overall processes. Introns are the first part. This is a piece of information on the DNA that is not turned into a protein. So an intron is what is sometimes called a junk portion of DNA or junk DNA, because we don't really know what it does. So we've done research on what enzymes are created, what proteins are made, all of the other things that DNA is doing. These portions have not been found to make or do anything specific inside the cell. So we call those an intron, so it doesn't create a protein. The exon is the portion that will be creating the messenger RNA strand and will thus then be creating a protein later on when it reaches outside the nucleus and grabs or attaches to the ribosome. It will be creating a protein. And so the last part of that, dealing with some of these terms, is a codon or a triplet codon. And a triplet codon is a sequence of 3 nucleotides, or 3 nitrogen bases, because that's the important piece of information that we're looking at, that ends up determining what amino acid will get added to the next chain, or to the chain of amino acids for the protein to be created. And so we'll be talking more about triplet codons tomorrow, but that is where we are going to stop today, looking at introns and exons, because that's the important portions of information you can see here that will create a messenger RNA strand. And so this messenger RNA strand will eventually be creating protein, but we will cover that in the next portion of notes.